Hi guys. Hopefully when you're watching this, I'm actually sitting right there in front of you. But as I probably already, if that's true, as I probably already explained to you, I am going to record this lecture just to try to save my voice today and use this instead of doing it live. As you can see, I'm still on oxygen and I still have issues with this cough. We'll see if it rears its ugly head as it just did. Um, you see, speak of the devil. There you go. See if it becomes problematic. Say it that way. While I try to go through this lecture with you. So, actually, let me just do this. You take the cough drop right now. I apologize for sucking on a cough drop while we're doing this, but hopefully that'll help me get through this. But you can see why, if I'm actually there live, I would prefer not to have to do this twice in one day and just do it the one time and use this recording instead of doing it live. So let's give it a go, shall we? So we're going to talk about the dogma of molecular biology today. And with that, let's go to the slides. If I can do this right. There we go. Let me just double check this. Okay, that should be right. So hopefully, there we go. Hopefully we're good now. So in any textbook you look at, any textbook that shows life sciences, they're going to show you this as the dogma of molecular biology, where DNA is used through the process of transcription to make RNA, and then RNA is used through the process of translation to make proteins, and DNA is used to make more DNA through the process of replication. So this is exactly what you will find in, like I said, any textbook on life sciences. But what I want to tell you is that this is incomplete. And we're going to get back to discussing why that is after we go through talking about these processes. So first of all, transcription is the process where DNA is used to make an RNA copy of itself. RNA stands for ribonucleic acid, and transcription is the first process we see in the dogma of molecular biology. Oh yeah, I wanted to show this short video. This guy right here.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Okay.
For just $67, you can make as many videos as you want, and you never need to pick up a camera or use.
Well, you got a free commercial. Well, partially a free commercial out of that, huh? All right, so back to this. So you got a little 3D animation showing you these processes. So let's move on. So through transcription, an RNA molecule is created that is complementary to a DNA sequence. RNA is a nucleic acid, just like DNA, but as I explained in the previous lecture, RNA nucleotides utilize ribose instead of 2' deoxyribose, which is used by DNA. And I'm just going to keep going through these at a regular pace, but I'll pause it in class as much as you need me to get caught up on the fill-ins. So this is what I showed you before. In RNA, on the left, they use a typical ribose sugar, which has the OH in the 2' position. And DNA on the right is missing that oxygen, only has the H, which is why it's without oxygen, or deoxy. So they call it deoxyribose, and that's what gives it its name of deoxyribonucleic acid, whereas RNA is just ribonucleic acid. And here is a DNA nucleotide showing the 3 groups and that 2' position having only the H, versus an RNA nucleotide where we have the same 3 groups, except that sugar becomes a ribose because it has the OH at the 2' position. So RNA has ribose incorporated into its backbone, not deoxyribose. RNA is typically single-stranded as opposed to DNA being double-stranded. And RNA contains 3 of the same 4 nucleotides as DNA, with the exception of T. RNA uses G, A, C, and U, not T. U is uracil, which has an almost identical structure to T, but it's missing a methyl group, a CH3 group, attached to the ring at the 5-carbon position. So if you look at the 4 bases of RNA, it uses uracil instead of thymine. The other 3 are just the same as they are in DNA. So in RNA, U takes the place of T in DNA. Oh, by the way, let me back up. So this would be the 5-carbon position. So that's where in the T, there would be a CH3 group there. And in uracil, you see it's missing that CH3 group. Sorry, I backed up there. So U takes the place of T. So wherever you have a T in DNA, you're going to have a U in RNA. So U is complementary to a T in DNA. So transcription is the first step in the process of creating the proteins encoded in the DNA. And this process is primarily accomplished through the function of an enzyme called RNA polymerase II. So DNA is not just A's, T's, G's, and C's that are haphazardly and repetitively placed throughout the molecule. There is order and logic of the placement of these 4 bases. And there are numerous reasons for the many arrangements, but for what we're going to talk about here, related to transcription and translation, we're going to focus on their arrangement into genes. So a gene is a unit of heredity, a functional region in the DNA sequence. A gene encodes a functional RNA molecule and contains all the regulatory aspects to ensure the RNA is transcribed accurately. So it's a stretch of DNA that's a template for an RNA, which is then used to be made into a protein. Genes encode the transcription of an RNA molecule, which is most often then translated into a protein. So generally speaking, genes encode proteins. And RNA is the RNA is the messenger from the DNA, which tells the cell machinery how to do that. Genes contain the DNA blueprint that tells the DNA, or that tells the cell machinery how to assemble a given protein. Via the messenger, the RNA. Now, an exception is that sometimes an RNA functions as an RNA, and it's not translated into a protein. But whether the RNA here we go. Hopefully this will calm down here. But whether the RNA transcribed you see why I wanted to save my voice in class and not have to do this twice? All right, let's try this again. Whether the RNA from the DNA is translated into a protein or remains a functional RNA molecule, it is transcription of a DNA gene that creates the RNA. So you can imagine DNA as an enormous template of the given order of the 4 bases. And through the process of transcription, the DNA template is copied into an RNA molecule that contains RNA bases in the identical order of the DNA bases. And the one difference, like we said, is that an RNA, U is used in place of a T. But to the process of molecular biology, they mean the same thing. So remember, DNA is 2 strands of sequences of the 4 bases, each running in the opposite direction of the other, and therefore they're each read in the opposite direction of the other. Remember this image? They're read in the 5' to 3' direction. So the strand on the left would be read going down. The strand on the right would be read going up. And then, like we said, the 2 strands are held together in the middle with the hydrogen bonds. Well, they have names for these strands, of course, right? It wouldn't let you get off that easily. The one strand's called the coding strand, also called the sense strand. And this strand has the same base sequence as the RNA that's produced. The other strand, the non-coding strand, or the antisense strand, is the strand on which the RNA is actually created. So remembering A always binds to T and C always binds to G, and vice versa, which means the 2 strands of DNA are complementary to one another. So RNA is created on and is complementary to the antisense strand of DNA. Therefore, it can hybridize to it, or it can bind to it and create an RNA-DNA double helix while it's being created, just like it was the other DNA strand. But the RNA is actually identical to the other DNA strand, the sense strand. So the RNA strand actually takes the place of the DNA sense strand, being identical to it, but it's created from the template of the DNA antisense strand. So if the left strand would be the one going down to the left would be the antisense strand, when you made your RNA, you would be making the bases that show up here. But they would bind to the bases over here, right? Except every time we'd have a T, we'd end up putting in a U. So the antisense strand would be the one. Hold on, I think I said that backwards. Let me start all over. Right? The RNA is identical to the sense strand. So let's start all over here again. So if the strand over here on the left going down, if that is the sense strand, it would be made with bases that bind to these guys over here, right? But it would be identical to these guys over here, with the exception that every time there's a T, we're going to have a U. That's what I meant to say. So, if we look at this process in a 2D cartoon, if here's our coding strand up here, here's our non-coding our antisense strand down here in the bottom. You can see as RNA polymerase is creating, the red would be the RNA, right? The RNA would match the bases in here, would match all these bases in here off of the DNA. Sorry, I can't draw too good. Except where there's a T, we're going to have a U. But all these bases are going to bind to all these bases on the DNA down here. So this red will be identical to the sense strand, but it will bind to the antisense strand. The genes are not the only DNA sequences that get transcribed into RNA. It's the gene structure, or the DNA itself, that tells the RNA machinery where to start, where to stop, and which genes to transcribe, that's what I'm trying to say, at what time. Now, us humans have between 23 and 25,000 unique protein coding genes. Before they sequenced the human genome, that was far lower, was a far lower number than what they expected to find. And in fact, only about 1.5% of the human genome codes for proteins, while the rest consists of RNA-only genes, regulatory sequences, repetitive elements, and non-coding DNA. There's also genes inside of genes, genes in both directions, overlapping genes, genes read in more than one reading frame. There's pseudogenes. There's all kinds of stuff in there. But only about 1.5% of it, of our genome, codes for proteins. As I said, transcription is primarily accomplished through the enzyme called RNA polymerase II. But he gets help from his friends, other enzymes that are called transcription factors. So we're going to go through some of those. So RNA polymerase II matches A on RNA with T and DNA, U on RNA with A and DNA, G with C and C with G, right? Because if this was DNA, this would be a T, right? Binding A. But remember, with RNA, we use U instead of a T. So the template DNA strand it reads is called the non-coding strand, or the antisense strand, right? The one to which the RNA is complementary to. It can bind to it. The DNA strand that it mimics by creating a strand that's identical to it is called the coding strand, or the sense strand. And that's the DNA strand that the RNA would be identical to, with the exception of U's where there would be T's. So RNA, created to be used to create a protein, is called messenger RNA, or mRNA. Put the little M in front of the RNA. The completed mRNA is subject to editing and modification. And prior to the completion of the editing and modification, it's called pre-mRNA. So back to this, right? And we'll actually come back to this in a minute, but I'm coming back to this now because we're going to move over here. We're going to talk about what happens to RNA after it's been modified and edited and it's ready to go as a messenger RNA. We move into the process of translation, creating proteins from the mRNA molecule. And translation is an appropriate term because the language of DNA provided through the RNA copy that's acting as the messenger, it has to be translated into the language of proteins. So it's definitely an appropriate term for this process. And this is accomplished primarily through a structure called a ribosome. Ribosomes are made of ribosomal RNA, where you put a little R in front of the RNA, and ribonucleoproteins. Just means they're made of proteins and RNA molecules that are combined together. So ribosomal RNA is an example of an RNA that's made from the DNA that does not get translated into protein. It's used as an RNA. Ribosomes read the mRNA sequence in a 5' to 3' direction, and they decode the information in the bases. So ribosomes are the Rosetta Stones of what's called the genetic code. They translate the information from the bases into the corresponding amino acids. You guys remember that joke I told you? I think I told you this one already. What do you call an acid with an attitude? Amino acid. Ha! I think that one's funny. Like nucleotides to DNA, amino acids are the building blocks of proteins. They're subunits that get linked together in varying orders to form specific proteins. There are 20 different essential amino acids that we use to create proteins, as opposed to, remember, there's only 4 nucleotides with DNA or RNA. So we have more building blocks when it comes to protein than we do nucleic acids. And amino acids share something in common with nucleotides in that they're also nitrogenous, meaning they contain nitrogen. The secret behind the ribosome's ability to translate the information in the bases into amino acids is through the use of something called codons. And a codon is a sequence of 3 adjacent bases that are read together as one unit. So a codon is 3 bases that function as one unit. Each 3-base codon codes for an amino acid, with the exception of 2 special categories that we'll talk about here in a second. We'll talk about it right now, actually. The first special category is a stop codon. They don't code for amino acids. Rather, they're a signal to tell the process of translation to stop. So we're not going any farther from this spot. So that's a stop codon. In DNA, those sequences are TAA, TAG, and TGA, talking about in humans here. Or in RNA, remember those T's become A's? It would be UAA, UAG, and UGA. Those would be the stop codons. So when a ribosome reads any of these 3 codons, translation is going to stop. It knows this is as far as we go. This is the end of this protein. The second special category is the start codon. And this is the signal that tells translation where to start. And the start codon in humans is the amino acid methionine. And in DNA, that's an ATG. In RNA, it's an AUG. So that's the signal that when a ribosome reads that start codon in the mRNA, translation is going to begin there. So a ribosome will go along mRNA looking for the start codon, and it will not do anything until it comes to the start codon. Here is a 2D image of this process. This is where I'm showing, see how we're matching 3 bases at a time here with 3 bases. And notice how the amino acid here hooked to the tRNA, 3 bases again, right? That will come in and be used in a codon. And as it gets placed in its appropriate spot where it matches up, then the amino acids get released and they get hooked into this growing chain. And then this uncharged tRNA will go to be recharged. It'll add its amino acid back to it. As I'm trying to show here, this mRNA gets fed through the ribosome, kind of like an assembly line. So it's fed through the ribosomes. As it does, the ribosome reads the mRNA, 5' to 3', and it decodes the information in the bases. And this whole process starts when it reads the start codon, AUG, which would be ATG in the DNA. When it reads the start codon, the large subunit recruits a transfer RNA that contains the complementary 3 bases called the anticodon that will bind to this codon. So for AUG, the complementary 3 base anticodon would be, yes? What would it be? UAC, right? UAC. Sorry, I can't write too good with this. UAC. A binds with U binds with A, and G with C, right? UAC. The transfer RNA molecules are covalently bonded to amino acids that match the amino acid called for in the codon. And that binding is facilitated by the enzyme called aminoacyl tRNA synthetase. That's a fun one to say, isn't it? At least one type of tRNA exists for each of the 20 possible essential amino acids. And in some cases, there's more than one. And like I told you already in that image, when it's complex with its amino acid, a transfer RNA is considered charged. The ribosome's smaller subunit binds to the messenger RNA, and the larger subunit binds the transfer RNA molecules linked to the amino acids. So here's the smaller subunit. So the messenger RNA would bind to this guy, and then all the tRNAs would bind up here to the large subunit. On one end of a charged tRNA is a 3 base anticodon that's complementary to the 3 base codon in mRNA. And when the appropriate tRNA is recruited, it's base paired with the mRNA 3 base codon. This occurs in what's called the A site of the ribosome. Oh, I thought I had another picture. I don't. Back up to here. So here's the A site. So it starts, as you see how this guy's coming in here? It starts in the A site. This is the first thing that's going to happen. This guy's going to come in and bind right in there. So this process also aligns the other end of the charged tRNA in the appropriate position for it to be, for its linked amino acid to be added to the protein being created. Within the larger subunit of the ribosome, the amino acid is released from the tRNA and added to the growing chain of polypeptides. And that happens in the P site. So right here, right? So see how when we get to here, this is going to be released and added to the growing chain. That's added by the peptidyltransferase activity of the ribosome. Peptide bonds are created that covalently link each amino acid to the previous amino acid in the chain. So the large subunit of the ribosome is responsible for doing this. The uncharged tRNA is released to be recharged with another amino acid of the same type. And that happens in the E site, right? Over here in the E site. So where he's being released, and he's going to, like I already said, he's going to be recharged with another amino acid. So a question you might have at this point would be, how does the ribosome know what amino acid each codon in the mRNA calls for? And here's where we need to return to something I previously mentioned, that being that ribosomes are the Rosetta Stones of what's called the genetic code. And they translate the information from the bases into the corresponding amino acids. And here, ladies and gentlemen, is the genetic code. And if you look through here, what we see is each of these three bases in the codon calls for a given amino acid, or it calls for stop codons. Here's methionine, which also functions as the first one that it comes to. When it's reading through the mRNA, the first one also functions as the start codon. But you can see all these three different base combinations code for a given amino acid. Some of them have several that code for it. For example, all of these code for proline. All of these code for serine. All of these code for leucine, and these are phenylalanine. But this is what is considered the genetic code, and this is how the ribosome matches the appropriate tRNA with its matching amino acid to the codon called for in the mRNA. So once the appropriate charged tRNA has been annealed, means it binds to its complementary codon, the ribosome moves along to the next codon. And this process just keeps repeating over and over until it reaches a stop codon. When it reaches the stop codon, the ribosome will fall apart and fall off the mRNA. So the two subunits, the large and small subunits, will disassemble. They'll come apart when this happens. So the process of recruiting the correct tRNA amino acid complex, lining them up, releasing their amino acids and setting them free, while peptide bonding each amino acid to the previous one, all of this happens similar to a conveyor belt moving through an assembly line, or like a ticker tape machine. But you guys are probably too young to even know what that is. But it was a machine they used to communicate. I used it a lot for stocks back in the day. And the little bitty tape was fed through the machine as it typed information on it, kind of like a conveyor belt moving through an assembly line. All right, back to this. The incomplete version of the molecular biology dogma. I already told you about that, right? That this was considered, or that this is incomplete. But the thing is, you're never going to read, when you see this information, like I said, in any of the life sciences textbooks, you're not going to read anything about it being incomplete. Which is why I'm going to tell you. This is the complete version of it. Here we still have DNA making RNA through transcription, RNA making proteins through translation, and DNA making more DNA through replication. But what they will not tell you, or you won't see in any of those textbooks, is that in order to make RNA from DNA, you need RNA and proteins. So you need RNA and proteins to be there before you make RNA and proteins. And you make DNA, and you need DNA to be there before you make more DNA. And you need RNA and proteins with existing DNA to make more DNA. So you need the one to get the others, but you need the others to get the one. And if you stop and think about that, that should make you scratch your head a little bit, wondering, well, that doesn't make sense. How is that possible? And that's the whole point. This entire process is scientifically impossible. And that's one of the reasons they won't tell you that the molecular dogma version that they show you like this is incomplete. Because they don't want you to know it's scientifically impossible. But there it is scientifically impossible. These processes that we just talked about in molecular biology require at least 100 unique proteins and about 200 unique RNA molecules. You need about 100 RNA molecules to create the 100 proteins. And then there's more RNAs that you use just as RNA. So there's about 300 molecules required for all these life essential functions of DNA. All of this, there's about 300 of these molecules required. And that's just to turn the machines on, so to speak, to get this system started. That's before you start making all the RNA and proteins that you need to make our cells and our bodies and all that kind of stuff, right? That's just to make this process work. But like I was just explaining, you can't have any of those 300 molecules without the DNA, and you can't have the DNA without any of those 300 molecules. You need the DNA to make all those molecules, but you need all those molecules to make the DNA. And on top of that, you need pre-existing DNA for all of this to happen. Which means they all have to be existing and functioning together at the same time, or none of this can happen. And if none of this can happen, life can't happen, because the molecular biology dogma is what life is based on. That tells us that all of those 300 protein and RNA molecules must all be present at the same time from the beginning, or you cannot have life. And like I said, this is scientifically impossible to explain. And they not only have to be there, but they have to be functioning together. They have to be doing their jobs, right? They have to be have the appropriate and necessary subunits to interact with one another and function together to make this system work. You see, the problem is all these subunits for DNA, RNA, and proteins, they will not self-assemble. They have to be assembled by other molecules. It cannot and will not happen any other way. They will not auto-assemble. And that's one of the reasons that everybody knows this is scientifically impossible. Even though you won't hear people talking about it that way, it's a fact that it's scientifically impossible. So you have to have all these molecules present and functioning together, or you can't have life. And the problem, does anybody see a problem for this? Is what I'm just saying. It's scientifically impossible. There are no theories, scientific theories, that can explain how this is possible to have come into existence. And that's why I told you that when you see the version of this in the books, this is all they're going to show you. They're not going to show you this because this is impossible to explain. But I wanted to make sure you knew that, so I wanted to explain that to you. And, ladies and gentlemen, I think that's the last one. It is the last one. Okay. So we will come back over here to this. And the rest of the class now, we will move on to the other stuff that I have. My camera's tilting. This camera's not so good. That's a little straighter, isn't it? Ah, good enough. So we will move on now. I'll give you the rest of the stuff that I have planned for you today. I got some handouts for you, and we will move on to that. All right? So I kind of made through without coughing too much. Had a few coughing fits in there, but I would say it survived pretty well. So let's move on to all the stuff we were going to be doing in class now.